Hello, I'm Juliet Strubel, and I will be talking about the 2020 status of blood-brain barrier opening for drug delivery. So to start with, I identified 227 preclinical and clinical articles which discussed blood-brain barrier opening for various neurotherapeutics. The reference list of these studies were examined and this uncovered additional articles. Many of the papers were dedicated to how focused ultrasound opens the blood-brain barrier, which substances can move across it, and what result comes from this. So starting with the neurovascular unit on the left, showing the many layers substances need to cross in order to reach the brain, starting with the inner layer, which contains endothelial cells with tight junctions in between them, the basement membrane, the pericytes, and then the astrocytes. So in between endothelial cells are tight junctions. One is shown in the diagram on the right here. It contains transmembrane proteins such as occludins, claudins, and junctional adhesion molecules, all of which inhibit paracellular transport. It's helpful to think of these transmembrane proteins as rubber bands which stretch when pressure is applied to them. As we've previously seen, A is a picture of the neurovascular unit, and B shows the transport mechanism used to cross the blood-brain barrier, such as paracellular transport, which is actually the most well-understood mechanism, and transcellular transport with numerous subtypes. So how does ultrasound actually open the blood-brain barrier? Well, this video shows the oscillation of microbubbles as ultrasound is applied, which puts pressure on the endothelial cells, therefore opening the tight junctions and then releasing the drug. And this method is advantageous because the delivery is completely non-invasive. It provides local and targeted disruption, which leads to high spatial precision. It's completely temporary, so it closes after 24 hours and the process is repeatable. There are three mechanisms by which we've already proven that ultrasound can help open the blood-brain barrier. The first is through tight junctions, which are opened by oscillation with the microbubbles, which shows that the transmembrane proteins have been reduced. The second is through transcytosis, proving that focused ultrasound increases the number of transport vesicles. And the third is through efflux proteins. So focused ultrasound decreases the efflux proteins. So blood-brain barrier disruption is directly correlated with reduced PGP intensity, so the drugs will not be pumped back out. There are potential dangers with blood-brain barrier opening in ultrasound, such as hemorrhages, ischemia, cerebral edema, direct injury to the cells, extravasation of substances, neuroinflammation, which is concerning because that can lead to neurodegeneration. So it's important that we get it just right. These are examples of getting it just right regardless of the mechanism. We've shown preclinically that increasing the pressure increases blood-brain barrier opening for larger molecules, and we've shown clinically that the greater the blood-brain barrier opening, more of the therapeutic can enter, resulting in a better outcome for the patient. In summary, the higher the pressure, the greater the enhancement, and the greater the blood-brain barrier opening. I used the chart on the left from 2015 as a start to classify the drugs in the table, and the diagram on the right is further proof of how beneficial focused ultrasound is, with MR-guided FUS producing a 30-fold increase in fluorescent cDDP BPN coverage in the tumor tissue. This is the compilation of all the literature for preclinical and clinical work on focused ultrasound-induced blood-brain barrier opening. Every different color represents a different source. This table is important because people are focusing on the best formulation for packaging for therapeutic delivery. So the drugs that are circled in red are the drugs that are actually currently being used in clinical trials. So it's also important to remember the first MR-guided focused ultrasound blood-brain barrier opening in humans showed that in areas of sonication there was an elevated concentration of therapeutic compared to an unsonicated tissue. So these are the current clinical trials taking place for blood-brain barrier opening with drug delivery for brain tumors. A good example of the relevance of drug packaging is lipodox, seen in the table. Packaging drugs in nanoparticles is actually much more beneficial, as these are very toxic drugs, so systemic toxicity is decreased. So liposomes are the most common developed type of nanocarrier, and there are two ways by which liposomes are able to deliver drugs to the brain. The original thought was that the liposomes release contents into the blood-brain barrier so that they can cross, but there is evidence here which shows that the liposomes themselves cross the endothelium, which is very beneficial because this allows for much more targeted drug delivery. This is a summary of the types of particles by sizes and weights, going from small to large. So you can see that we need to open the blood-brain barrier to a greater degree when using larger molecules. For example, temozolomide compared with a smaller chemotherapy agent. To summarize, I have discussed the 5 layers of the neurovascular unit. I've talked about how ultrasound opens the blood-brain barrier and by which mechanisms these drugs can pass through. I've talked about the various classes and sizes of therapeutics that have successfully crossed the blood-brain barrier with ultrasound, and I have presented a summary in an illustration. We've shown that preclinically many mechanisms are helpful, so there's no shortage of diseases where we need to improve drug delivery. So we have ALS, Alzheimer's disease, Parkinson's disease, diffuse intrinsic pontine gliomas, stroke, and Huntington's disease. For future studies, we need to better understand how to control the extent of blood-brain barrier opening and maybe perhaps control the mechanisms by which drugs can pass through, and also think about, are certain mechanisms safer to open than others? Finally, I would like to say a huge thank you to Dr. LeBlanc for her mentorship, to Dr. Kelsey Timby for her involvement with the table, and to the Focus Ultrasound Foundation for their support.